Here is an interesting question. A test cross is performed in a pea plant producing yellow-colored seeds. The offspring produced from the cross had 50% plants producing yellow-colored seeds and 50% plants producing green-colored seeds. It can be inferred that the genotype of the parent pea plant producing yellow-colored seeds is. Now, before I look at the options, let me first list out all the information that I can gather from this question. It is given that a test cross is performed, right? A test cross is performed in a pea plant that produced yellow-colored seeds, right? Also, it is given that in the resulting offspring, it was observed that 50% of the plants produced seeds that were yellow in color, and the remaining 50% of plants produce seeds that were green in color. Now, by making use of this information, we have to figure out what is the genotype of this parent pea plant. Okay, we know its phenotype, we know that it's going to produce yellow-colored seeds, but we don't know its genotype. Whether it is homozygous, heterozygous, dominant or recessive, that we don't know. That is what we are required to figure out by making use of this information. Now, let's look at the options. Option A, homozygous dominant. Option B, heterozygous dominant. Option C, homozygous recessive. And option D, heterozygous recessive. Okay, so in this question, the character of the pea plant that is considered is the color of the seed. In pea plants, seed color can be of two types. There are two variants of seed color, which we call the two contrasting traits. Seed can either be yellow in color or it can be green in color. The yellow color seed is considered to be dominant over the green colored seed. Okay, so if we can consider the allele that is responsible for the production of yellow color seeds as capital letter Y and the allele that is responsible for the production of green color seeds as small letter y, since the yellow seed trait is dominant, the genotype of this plants that are yellow in color can either be homozygous, meaning it can be capital Y, or it can be heterozygous. Okay, but if it is recessive, it is always going to be homozygous. Now in this question, what is it that we have to find out? We have to find out whether the pea plant that produced yellow colored seeds, the one that was test crossed, whether it is homozygous dominant or heterozygous dominant. We already know that if it's producing yellow color seeds, it is dominant. So the two options that had recessive, that can be right away eliminated. We are only left with two options. One is homozygous dominant, one is heterozygous dominant. And that is what we are required to figure out. Okay, so how exactly can you find out the genotype of a plant that is showing the dominant phenotype? So that can be figured out by performing a test cross. What exactly is a test cross? A test cross is a cross that is performed to predict the genotype of an organism that is showing the dominant phenotype for a specific character. So how exactly is a test cross performed? In a test cross, we take the plant that is showing the dominant phenotype. Okay, in this case, it is the plant that is producing yellow colored seeds. So it has the dominant phenotype, but we don't yet know about its genotype. This cross is being performed in order to find that out itself, right? So this plant, we will not know the genotype, so I will write unknown genotype. A plant that is showing the dominant phenotype with an unknown genotype is crossed with a plant that is showing the recessive phenotype. Okay, so this cross is what is called the test cross. In the question, it was given to us that in the resulting offspring, there was a 1:1 ratio of plants that produced yellow colored seeds and green colored seeds. Okay, so in order to obtain a ratio of 1:1, that is possible only when the parent plant that is producing yellow colored seeds is heterozygous. Okay, remember, we don't know its genotype. It could be homozygous or heterozygous. But what I'm trying to tell you here is, if you have obtained 1:1 ratio in the offspring, that is possible only if the parent plant that is producing yellow colored seeds is heterozygous. Here's a cross. Okay, so if one of the parents, if the parent is capital Y, small y, if it is heterozygous, then it will produce two types of gametes, right? Capital Y and small y. We are crossing that with a plant that is showing the recessive phenotype. So its genotype obviously will be homozygous recessive, and this will produce gametes that will contain the allele small letter y. As you can see in this resulting cross in the offspring, you can see how 50% of the plants, these two among the four, have the genotype capital Y small y. Whereas the other 50%, 2 of 4, have the genotype small y small y. So the genotypic ratio in this cross is 1:1. So is the phenotypic ratio. Okay, you can see how these two plants are heterozygous dominant. They will be producing yellow colored seeds, and this homozygous recessive genotype plant will be producing green colored seeds. The phenotypic and the genotypic ratio, if it is 1:1, then it is obvious that the plant that was crossed is the plant that was test crossed was heterozygous dominant. Okay, so now we know that the parent plant that was producing yellow colored seeds, it had the heterozygous dominant genotype, which is why we could get the ratio of 1:1 in the resulting offspring. So the correct answer to this question is option B, heterozygous dominant. Here's the question. A genetically dwarf plant starts growing and becomes a tall plant after the application of plant growth hormones. This genetically dwarf plant was crossed with a pure line tall plant, and the resulting F1 hybrid was self-pollinated. What will be the phenotype of the F2 generation? Now, this is a very long question that has a lot of information. So before I look at the options, I'm just going to write down all the information that I can get from this question. So it is given that there's a genetically dwarf plant. This dwarf plant, it grew tall after the application of plant growth hormones. Now, this genetically dwarf plant is crossed with a tall plant, which is, it's also mentioned that it is a pure line. And as a result of this cross, the offspring that is obtained is referred to as the F1 generation, right? Now, this F1 generation plants is allowed to undergo self-pollination. The offspring that is obtained as a result is the F2 generation. Now, what is it that we have to find out in this question? We have to find out what is the phenotype of this F2 generation. Okay, now let's look at the options. Option A, all will be dwarf plants. Option B, all will be tall plants. Option C, 50% will be dwarf and 50% will be tall. Option D, 25% will be dwarf and 75% will be tall. Now in the question, it is given that it is a genetically dwarf plant that could grow after the application of plant growth hormones, right? Just because it grew tall because of application of hormones, it does not change the genotype of the plant. It will still carry alleles that are responsible for dwarfness in the plant, okay? So the genetically dwarf plant will still have small t as the alleles, and the gametes that are formed by this plant will also carry small letter t allele for the character height. Now, when this dwarf plant is crossed with a pure line tall plant, what is a pure line tall plant? Pure line tall plant is a tall plant that is homozygous. Okay. So, since it is homozygous, it will carry capital T as the genotype. The gametes that is produced by the tall plant will carry the allele that is responsible for the tallness. Okay, so it will carry capital T as the allele. Here we are crossing the genetically dwarf plant that has the genotype small t with a tall plant which is a pure line. It will have the genotype capital T. So the tall plant will produce gametes that will have capital T allele, and the dwarf plant will produce gametes that will have small t as the allele. Now, as you can see, all of the offspring, 100% of the offspring will have the genotype capital T small t. Now, capital T small t genotype will have the phenotype tall, right? It is heterozygous dominant and it will exhibit the dominant phenotype. So all of the 100% of plants, offspring that are produced by this cross, the F1 hybrids, they will be tall. Now, the F1 hybrids are allowed to undergo self-pollination. So on selfing of F1 hybrid, these plants are allowed to undergo self-pollination. So both the parents here will have the genotype capital T small t, right? So two types of gametes will be produced. Each parent will produce gametes, half gametes that will carry the allele capital T and 50% gametes that are carrying the allele small letter t. Okay, now the offspring that is produced as a result of this cross is called the F2 generation. Here you can see how the genotype, there are three types. There's one here, capital T, homozygous dominant. There are two here, there are heterozygous dominant, and there's one here that is homozygous recessive. Okay, so the genotypic ratio will be 1:2:1. In the question, we were asked about the phenotype and not the genotype, right? So let's look at the phenotype. Capital T will be tall. This also is tall. This also is tall. And this will be dwarf, right? So the phenotypic ratio of the F2 hybrid in this F2 generation offspring in this plant would be 3:1. There are 3 tall plants and 1 dwarf plant. So the number of dwarf plants out of 4, there was just 1, right? So 1/4 of the offspring was dwarf. So if you're calculating the percentage, 25% of the offspring is dwarf, and the remaining 75% of the offspring, 3/4 of the offspring will be tall. So 75% of the plants will be tall and 25% will be dwarf. So the correct answer to this question is option D. 25% will be dwarf and 75% will be tall. Here is an interesting question. A pea plant producing round seeds is crossed with another pea plant producing wrinkled seeds. In the F1 generation, 50% of the offspring produced wrinkled seeds. Predict the genotype of the parents. In the options, we have capital R crossed with small r, capital R crossed with capital R, option C, capital R crossed with capital R, and option D, capital R crossed with small r. So we have to find out what is the genotype of the parents that are involved in this cross. Now the character that is considered in this particular cross is the shape of the seed of pea plant. In pea plants, the seed shape can be of two types. Either it can be round or it can be wrinkled. Now these two are called the contrasting traits for the character seed shape in pea plants. Among these two, it is the round seed shape which is dominant over the wrinkled seed shape. Remember, the dominant trait can exist either in its homozygous or heterozygous form, which means the round seed shape producing plant can have the genotype capital R, homozygous dominant, or it can be heterozygous dominant, capital R, small r. Whereas if it is showing the recessive phenotype, then it will have to be homozygous. So if one of the parents is wrinkled, it is obvious that it will have the genotype small r. Okay, so this is small r, and this can be capital R, or it can be capital R, small r. Okay, so there are two options in this question with which do not have small r as one of the parents. Okay, one of the parents have to be small r, because wrinkled seed shape producing genotype is small r itself. So there are two options here that do not have small r as one of the parents, and we can right away eliminate that. Now we are left with two other options that have small r, the homozygous recessive genotype as one of the parents. So that is what we are going to consider now. Okay. Now let us consider the cross that has the dominant homozygous dominant genotype and the homozygous recessive genotype. In such a cross, you can see that all of the resulting offspring, all of them will have the phenotype round. As you can see, all of them are capital R, small r, which is the heterozygous dominant genotype. And all of them will have the phenotype round. But in the question, it was given to us that the resulting F1 hybrid also showed 50% wrinkled seed producing plants, right? So it is not possible for the parent to be capital R and small r. So the only other option that we are left with is capital R, and small r as the parents. If one of the parents, the parent that is producing round color seeds, round shaped seeds, has the genotype capital R, heterozygous dominant, then two types of gametes can be produced, one containing the dominant allele and one containing the recessive allele. And the other parent, obviously, if it's producing wrinkled seeds, then it will have to be homozygous recessive. It will produce just one type of gamete that will carry the recessive allele. So in this offspring, you can see how you get plants that are producing round seeds as well as plants that are producing wrinkled seeds. So as you can see, among 4 2 are producing round seeds and 2 are producing wrinkled seeds, which essentially means that 50% of the resulting offspring is producing wrinkled seeds. Since this is in line with what is given to us in the question, the correct answer to this question should be option D. If the plant that is producing round seeds has the genotype capital R small r, and the plant producing wrinkled seeds has the genotype small r, that is when you can get the F1 offspring that has 50% of plants that are producing wrinkled seeds. So the correct answer to this question is option D. Now let's take a look at this question. A recessive allele differs from a dominant allele with respect to the options are sequence of nucleotides, functionality of the encoded polypeptide, amount of functional polypeptide or enzyme, option D, all of the above. Now, in order to answer this question, we must first be aware of what a recessive allele and a dominant allele refers to. I want to begin by talking about the gene. Gene, I'm sure you know, is considered to be the basic unit of heredity, right? So what exactly is a gene? Gene is a specific sequence of nucleotides, usually in DNA in most organisms, except for a few viruses that have RNA as a genetic material. Apart from that, in most organisms, DNA is the genetic material. And a specific sequence of the deoxyribonucleotides in DNA will contain information for coding a protein. So it will contain information that specifies the production of a functional polypeptide or an enzyme. Okay, so that is a gene. Certain segments of DNA contain information for the formation of proteins or polypeptides. Those are called as genes. Okay, so there are different forms of genes that can exist, and alternate forms of a gene is what we call as alleles. So alleles can be defined as the alternative forms of a particular gene. Every character in an diploid organism will contain 2 copies of a gene. Okay, so every character is controlled by alleles, and in an organism the alleles can be of the same type or it can be of different types. Okay, so in the pea plant experiments that Mendel conducted, for the characters that he selected, he had 2 different alleles that are specifying the contrasting traits for every character. Okay, so whenever an organism contains 2 different alleles, a pair of dissimilar alleles, which we call the heterozygous condition, in such a case, only one of the alleles will be able to express itself while the other one fails to express itself. Okay, so of the 2 alleles, whichever one has the ability to express itself even in the heterozygous condition, that kind of allele is referred to as the dominant allele, and the one that fails to express itself in the heterozygous condition is what we call the recessive allele. For example, one of the characters that Mendel considered in his experiments with pea plants is the color of the seed. Okay, the seed color can be of 2 types, either it can be yellow or it can be green. But the yellow seed color is dominant over the green seed color. So the yellow colored pea is called the dominant trait and the green color one is called the recessive trait. Okay, so this yellow colored allele can express itself even in its heterozygous condition. Now, what exactly is the difference between a dominant allele and a recessive allele? So the difference between a dominant and recessive allele lies in their composition. What exactly does that mean? It means that the sequence of deoxyribonucleotides in their DNA will be different. So they have difference in their nucleotide sequences. Because there is a difference in their nucleotide sequences, the information essentially is different, right? So because there is a different kind of information, it gets reflected even in the polypeptide produced. Even in the sequence of the polypeptide produced, the sequence will be different. The recessive allele may produce partially functional, semi-functional, or completely non-functional polypeptide. Apart from that, the amount of polypeptide that is produced can also differ based on the differences in the information carried by the dominant and recessive alleles. These are the differences between the dominant and recessive alleles. So in the question we were asked, what is the difference between dominant and recessive allele? They differ from each other in with respect to the sequence of nucleotides, functionality of the encoded polypeptide as well, like I told you, and even the amount of functional polypeptide or enzyme. So the correct answer to this question is option D, all of the above. Now let's take a look at the question. What is a phenotype? Option A, the genetic makeup of an individual. Option B, it is the study of cells present in our body. Option C, it is an account of physiological activities. Option D, observable characters of an organism. Now this is a very easy question. What is a phenotype? It is the observable characters of an organism. For example, when you consider pea plants, the height of the pea plant is something that is easily observable, right? When you look at the pea plant, you can say whether it is tall or dwarf. When you look at the color of the flowers, you can say whether it is violet or white. So these easily observable characters in an organism is what refers to as what is referred to as the phenotype. Now, what determines the phenotype? The phenotype of an organism is determined by its genotype and also by its environment. What is a genotype? Genotype is the genetic makeup of the individual. For example, when we say a pea plant, talking about its height again, it could be tall or it could be dwarf, right? Whether it is homozygous or heterozygous dominant or recessive, that is what is the genotype of the tall plant that is present within every one of the pea plant's cells in its DNA, right? So that is the genotype which you cannot observe from the outside. So it is usually represented by means of letters. If it is a dominant allele, then it's represented as the capital letter generally. If it is recessive, it's represented as the small letter. So since in pea plants, the tallness is dominant over dwarfness, you have capital T, capital T, capital T, small t, which are genotypes for the tall pea plant, and small t is considered to be the genotype for the dwarf pea plant. Okay, in the question there was a mention of the study of cells present in our body. Now the study of cells present in our body is referred to as cytology, and the study of cells is called cytology. One option was account of physiological activities. Study of physiological activities or life processes is what we refer to as physiology. Okay, so both these options are incorrect. Here we were asked what is a phenotype. Now we know that the correct answer is option D. Phenotype refers to the observable characters of an organism.